The final function of DNA is protein synthesis. And that phrase kind of explains everything if you understand the word synthesis, to synthesize, to create, to make. So DNA makes proteins, makes all the proteins in your body. And the other thing you got to learn in this class is proteins aren't just muscles and bones. They are, they are the cells, parts of the cells that your nervous system uses. So these are the receptors, which we'll learn about in the next few lectures. Receptors where chemicals bind to. And if those proteins on the surface of those cells in your brain aren't working properly, well, you can have a host of different problems. So we have to understand about genetics and DNA so we understand how to figure out how chemical changes work in the brain. So protein synthesis is a two-step process. There's something called transcription and translation. So the directions for a protein are in a gene. So remember, we learned a gene is part of this DNA. Okay, and so when the message comes in that says, hey, let's turn this gene on because we need to make this protein, a process that's a little bit like replication but not the same happens. So in replication, the entire DNA is unzipped and a perfect copy is made. In transcription, the first part of protein synthesis, just the section of the DNA that holds the gene is unzipped. Okay, so it's broken apart from the double helix and the base pairs are separated. So now what happens during transcription, another enzyme, RNA polymerase, adds the complementary nucleotides to that strand that is open. And in this way, we go from making DNA, to making an RNA strand, what's called a messenger RNA strand. And then once the messenger RNA strand is made, it breaks off from the DNA closes back up, and now it's like a little worm, and it works its way out of the nucleus and comes into the cytoplasm of the cell. And then it's the ribosomes here that read what this message says. And so the message explains how to build the protein, which amino acid to put in which order. And so we say that the RNA now is translated from the messenger RNA to the actual protein. So protein synthesis goes from transcription, DNA to RNA, and then translation, RNA to protein.